Welcome to our antibacterial resistance project. My name is Nadine Borrero, and I will be presenting this topic with my partners Angelina de la Vega, Alina Amaro, and Alexandra Moya. We are going to go over four main key concepts. What is antibacterial resistance? How does it work? How do we test it? And finally, why is it important in biology and daily life? So, what is antibacterial resistance? Well, there are good and bad bacteria found in every organism. Antibiotics, which are drugs or medicines, are meant to fight off and treat harmful bacteria. However, resistance against antibiotics forms when bacteria gets accustomed or learn to fight against said drug. To test whether antibiotics are still effective, antibacterial resistance experiments can determine which bacteria have developed resistance against these different kinds of antibiotics.
How does antibacterial resistance work? Bacteria can develop antibacterial resistance through random genetic changes called mutations that can make a specific antibiotic ineffective. These mutations can be passed on through reproduction or direct contact with other bacteria. There are two main types of bacteria, gram-positive and gram-negative. Bacteria can be classified as either of the two using the gram stain, which will be discussed later in the video. It should be noted that nearly all bacteria have a cell wall made of a large-sized molecule called peptidoglycan. Pictured here is the peptidoglycan cell walls of gram-positive and gram-negative bacteria. Looking at the left, gram-positive bacteria have a thick peptidoglycan cell wall. Meanwhile, on the right, gram-negative bacteria have a thin peptidoglycan cell wall, squeezed between two membrane layers. For reference, in the images, the peptidoglycan cell walls are highlighted in orange. The differences between the cell wall structures affect how gram-positive and gram-negative bacteria react to the gram stain, which we will explain right now. The process of gram staining can be explained in 3 steps. First, the bacteria are dyed purple. Then, the bacteria are washed with a dye remover. And finally, a second pink dye, known as a counterstain, is added. Here is where that peptidoglycan comes in. The dye used in gram staining detects peptidoglycan. Gram-positive bacteria will remain purple because it has a thick peptidoglycan cell wall. Gram-negative bacteria will become pink because it has a thin peptidoglycan cell wall, and the outer membrane protects it. Now that a general idea of the cell wall structure of both gram-positive and gram-negative bacteria has been established, we can discuss how the cell wall structure affects the antibacterial resistance of these two types of bacteria. Gram-negative bacteria has a greater antibacterial resistance than gram-positive bacteria because of its cell wall structure. Gram-negative bacteria also develop antibacterial resistance quicker than gram-positive bacteria.
So how do we test it? Well, we test it by using the Kirby-Bauer test. So this test is set up with petri dishes that are filled with agar, which is a gel-like substance that creates a surface where bacteria can grow and be observed clearly. A thin layer of bacteria is then spread across the surface of the agar, and small paper discs are dipped into various antibiotics that are then spread out in different sections of the agar. Separating the paper disc is a crucial step in testing how much of an effect the antibiotic has had on the bacteria. This is because as the antibiotic fights off the bacteria, clear circles are formed around the paper discs. As you can see here on the picture shown right next to it, those clear circles are surrounding it.
These areas are called the zone of inhibition, or ZOIs. ZOIs are measured in millimeters and determine how antibacterial a specific antibiotic is against a certain bacteria. If the ZOI is less than 6, the antibiotic is not antibacterial. If the ZOI is greater than 6 but less than 16, the antibiotic is slightly antibacterial. If the ZOI is greater than 16, the antibiotic is highly antibacterial.
What is the role of antibiotic resistance in biology? It's pretty much how germs change to survive, so basically it's like evolution but for germs. So when we use antibiotics, most germs die, but a few tougher ones stay and live, and these are just as difficult to kill. And they can even share their survival skills with other bacteria. So scientists study this, and by studying it, They learn how bacteria can change and adapt, and pretty much it helps to understand how infections can spread and evolve. Overall, antibiotic resistance helps scientists see how life changes over time. So it's pretty much like watching evolution, but in tiny living things. What is the role of antibiotic resistance in real life? Basically, it's not something that happens in labs, it happens every day in real life. So if bacteria were to stop responding to medicine, infections that used to be a lot easier to treat, like ear infections, UTIs, and strep throat, could become really dangerous again. People might stay sick a lot longer or need medicine, and things like surgeries, chemotherapy, or organ transplants would become a lot riskier since they all rely on antibiotics to prevent infections. Basically, without antibiotics that work, everyday medical care would become much harder and scarier. Thank you so much for listening to us talk about
Antibacterial resistance.
all citations are referenced under the corresponding figures.
Here are additional works cited.